I am quite interested in metagenomics, microbiology, and astronaut health, astrobiology, all of those things. And I'll give a short introduction on how I got interested in these things. And then I'll talk about my project that I conducted during my internship at Pine Biotech. So, and overall is that my research was based on the adverse effects of high LET radiation on the mouse gut microbiome and consequently on the nervous system, causing psychological disorders like anxiety and depression. So why am I so interested in microbes? And it's because when we think of microbes, we usually think of pathogens, but that's changing now. We don't only think of microbes that make us ill, but we also think of them as a treatment now. We can help use these microbes using metagenomic studies to find unconventional treatments for diseases like cancer or atopic dermatitis. And also because it's obviously a core of astrobiology studies, and it's essential in understanding evolution because we were coexisting for all these years. So we cannot understand our own evolution without the evolution of microbes. And of course, as Dr. Gary and as his discussion touched upon it, in climate change and agriculture studies and finding renewable resources, microbes are invaluable. So I chose this topic specifically, astrohealth, because of the prospects of space travel and how they advance. Therefore, the research involving space travel increases proportionately. So among biology, biotech, and human research conducted on the ISS, the International Space Station, are studies on how humans and animals are affected by space travel, which will be important for us in the future. So it's a fast-paced and growing field, and my research specifically is on a space-type radiation called ionizing radiation, and more specifically, high LET, linear energy transfer radiation. High LET radiation can penetrate into microstructures and has enough energy to cause chemical change, which leads prominently to cell death. And according to the information collected by the ISS research, the effects of radiation exposure on human health are currently uncontrolled and will continue to be so for the foreseeable future. One promising factor, however, is that host microorganism interactions will be optimized, to say the least, by around 2028. So here's my methodology. I had three main stages, which was data exploration, statistical analysis, and then drawing conclusions. In the data exploration stage, I found my data set, which was on mouse gut microbiota from NCBI, and then using the Bioinfo platform, I used the Data2 pipeline and the phyloseq pipeline on the data set that I had, which was 16 as amplicons, to find paths that could lead me further into understanding the data. So then I conducted a NOVA and t-test to find if the observations I made during the phyloseq stage were correct. And then finally, I tried to connect these results to palpable therapy ideas, potential therapy ideas. So my data set was a total of 80 samples, and it was divided into two categories. One was based on radiation, going from 0.1.25 and 1 GY radiation, and the samples were collected 10 days post-radiation and 30 days post-radiation. And here is one of the outputs that I got from the phyloseq pipeline, which is NMDS, non-metric multidimensional scaling, which helps us observe the whole data set and find relative connections between them or separations. And in this one, we can see the relative separations between the families of bacteria found in those samples collected from the gut microbiota. And here I observed from this angle that 10 days post-radiation is more grouped than 30 days post-radiation. And then another output of the phyloseq pipeline was the Shannon index graph. And the Shannon index is a value used for measuring the richness and evenness of species in different samples. So from my comparison here, I found that between the the 4 different radiation doses, the Shannon index decreases with exposure to radiation at lower doses than at higher doses. And I also, during my research, found out that this was due to the properties of high LET radiation. And this is an excellent example that summarizes my entire findings during the data exploration stage. And from here, we can see from this phylogenetic tree that the OTUs that are significant, that change significantly from the control group, which was 0 GY. And we can see that 0.1 GY had lower abundances compared to 0. We can also see that OTU 4 and 5 have a common ancestor in recent times. So this is one of the most useful graphs that came out of the PhyloSeq pipeline that helped me make analytical observations. And then I decided to use statistical analysis, which was the ANOVA and t-test, to make sure that my observations were correct. And you can see from the simplified heat map that OTU 3, 4, 5, and 8 were the significantly decreased ones. And I also came to the conclusion that 0.1 GY collected at 10 days post-radiation was a significant group. And I also found out that there was a connection between the samples collected at 10 days post-radiation and the radiation dosages. So there was no relation between those two categories. And the most significantly decreased ones were the ones that were exposed to 0.1 GY radiation. So from this, I also came to the conclusion that the abundances for Lactobacillus, Bifidobacterium, and Erysipelotrichobacteria were the most significantly decreased abundances. The ones I'll be focusing on, however, is Lactobacillus and Bifidobacterium, since they contributed to the imbalance in the nervous system through protein coding genes that they have. And these protein coding genes correspond to GABA. And GABA is gamma aminobutyric acid, which is the chief inhibitory neurotransmitter in the central nervous system. And it is generated by Bifidobacterium and Lactobacillus too. Therefore, a decrease in these species of bacteria can cause or enhance disorders like depression and anxiety, which are very common in astronauts. And here you can see that there are two pathways, the GS and the GLS, through which these bacteria affect GABA. So from all of this, we can conclude by saying that lower doses of high LET radiation have a greater effect on OTU abundance. In this case, it is 0.1 GY. And we can also surmise that the acclimatization period is between 10 and 30 days, since the samples collected 30 days post-radiation did not pass the significance threshold. But we also have to keep in mind that these mice were not exposed to radiation in the long term, the way astronauts are. And some of my limitations were, of course, the difficulty of the taxonomic profiling, since 16S regions are quite similar for bacteria in the same genus. And then there's also the technicalities of optimization, and the limited sample size was another issue. But all in all, this is a good precursor study for microbiota studies in space. And further research should include synergistic effect of radiation and microgravity together. And of course, in this whole stage, I don't know, because I didn't have a bioinformatics background, I don't know how I could have achieved this if I hadn't come across Pine Biotech and the T-Bioinfo platform, because that greatly helped me, along with the advisors like Dr. Mohit, Ilya, and Dr. Harpreet, who helped me throughout my research. So here are my references, and thank you for listening to me. I hope this was enjoyable.
Thank you so much, Dilara. That was an amazing presentation, and it's a pleasure to listen to you every time. Well, there would be one question that we have in the chat, and it would be great if you could address it in brief. For example, first of all, what was the duration of the study, the samples that were exposed to the radiation? What was the duration? And finally, if you could explain the results again, and their like the phylogenetics that you got, and what those families usually you can conclude from those families that you could find.
Yeah. So basically, the duration was not specifically mentioned in the article that I chose the samples from, but it mentioned that it was during 1 day. So it wasn't as long term as astronauts do. And we have to consider in animal studies, of course, like, um, we can use mice, musculus, as a sample animal model, but we cannot expose it to as much radiation as humans are exposed to in space also. And I guess this was the phylogenetic one that you would like me to explain. So basically, when you run your samples through the phyloseq and data2 pipeline, they go through all the usual stages like normalization, and your samples are cleaned, etc. And then this is the output that you get. And from this, you can here understand that 1.1, 0.25, and 0. I don't know if I enlarge it, if you can see it more clearly. But from this, you can tell the abundances of OTUs, and OTUs are operational taxonomic units. When you run the Data2 pipeline and the phyloseq pipeline together, you don't exactly get all of the genus and species names of the bacteria that are in the sample. First of all, because there are a lot of DNA samples that are not titled, that are not named. And then there are also the fact that when you run these DNA samples are very volatile and they break a lot. So estimations have to be made, and the system basically, when you run the pipeline, will align the DNA samples that are present in these microbiota fecal samples as much as possible, and then it will not give it a name. It will not, first, it won't run it through the databases and say like, this is lactobacillus or this is bifidobacterium. The first thing you get is operational taxonomic unit, which is similar to saying this is one type of bacteria, but it's not specifically named. So that's what OTU means here. That's what the OTUs are. And then what you get is these correspondences, OTU4 is, and OTU5 is Bifidobacterium genus, and etc. And you don't usually get down to the species name if you don't optimize it too much, because of the limitations, as I said, of the numerous types of bacteria that are still not named or sampled. They're not in the database of these DNA samples, so they're usually left down to the genus name. So that's why that's how I made the correlation between OTU and the Bifidobacterium and Lactobacillus. Basically, I took out what was in the phyloseq pipeline, what this gave me as a correspondence. It gave me Bifidobacterium and Lactobacillus, and then I ran it again on the JGI database, and I found the genus and species name together, which are these ones.
That's
so
great.
That's
And
the phylogeny.
yeah, and is there a correlation between the abundances that you found and the neurological activity with GABA that you mentioned?
Yeah, so basically, because I was using samples that were previously, collected by other researchers, this was like a totally bioinformatics study, I couldn't exactly go back and analyze the behavior of mice. So I couldn't conclusively say that this decrease affects the nervous system. I can't say that because I didn't work with those mice, basically. But what I can do is run through the JGI pipeline and find that these bacteria have protein coding genes for sure that affect the human nervous system. They basically, through the central nervous system and through the interdendritic nerve, which is a nerve that goes through your gut and collects these chemicals, and it affects your central nervous system in return. So theoretically, I can say that a decrease in the abundances will cause anxiety and depression because GABA conclusively decreases. So that's what I